Hi everyone, thank you for joining me today. I'm Julieta Ramírez, analytical lead from INC and collaborator of UDA. I'm excited to discuss our research on the Zika virus and its complex interaction with vitamin D in encoding RNAs and the immune response in macrophages. Zika virus infection presents significant global health risks, particularly with its association with congenital malformations and neurological issues. Recent studies highlight the importance of innate immunity against Zika. Our study delves into how vitamin D can modulate immune response in Zika virus-infected macrophages, focusing on the roles of microRNAs and low-non-coded RNAs to construct a regulatory network and explore molecular interactions. Our objectives were threefold. The first one is to extract expression profiles. The second one is to construct a competing endogenous RNA regulatory network. And the third one is to explore the specific regulatory pathways involving non-coded RNAs and mRNA, emphasizing vitamin D in immune modulation during Zika infection. In this study, human monocytes were cultured and differentiated into macrophages with or without vitamin D. On day 6, MDAs and D3 MDAs were infected with the Zika strain. Then we extract total RNA and prepare miRNA libraries from samples. Following sequencing, we perform red alignment, followed by differential expression analysis. Additionally, we conduct BDR motif analysis, gene enrichment, miRNA target prediction, and build a competing endogenous RNA network. Okay, let's start with our first result on vitamin D modulation of lncRNAs in Zika MDAs. Building on our previous findings about vitamin D efficacy, we investigated the regulatory roles of lncRNAs. In our study, we analyzed 403 lncRNAs. These were clustered based on the expression patterns. For upregulated, we observed 6 clusters. Noteworthy examples include in cluster 1, this one is linked to cell adhesion, and in cluster 2, this one is linked to inflammation and stress, cellular stress. Among the downregulated cluster, in cluster 7 is we have here an immunomodulator, while in cluster A we have this one plays a role in copper inducer cell death. Next, we explore how vitamin D modulates cis-acting lncRNAs to regulate gene expression. We calculate Pearson correlation coefficients to identify co-expressed lncRNA-mRNA pairs, finding 129 interactions. Refining our criteria to include pairs within 300 kb of each other, we identify 15 significant lncRNA coding gene pairs. Relevant pairs include those that are here, which are linked to transcriptional regulation and viral pathogenesis. Additionally, we found a significant positive correlation between vitamin D exposure and the expression of these pairs, suggesting regulation via the BDR signal pathway. Next, we examined the expression levels of miRNAs across the experimental groups. We found 857 human mature miRNAs expressing in all the groups. In our analysis, we identified 23 differentially expressed miRNAs, 14 up and 9 down. This means clustering revealed 8 expression clusters, with cluster 1 to 4 showing the upregulation and cluster 5 to 8 showing the downregulation in response to vitamin D. Okay, further investigation into the BDR's roles revealed its binding to the gene loci of those miRNAs, suggesting they are direct targets regulated by BDR, which is crucial for modulating miRNA's expression patterns in Zika D3 MDMs. To assess the functionality of miRNAs modulated, we identified predicted mRNA targets for 17 of the 23 differentially expressed miRNAs, resulting in 78 miRNAs-mRNAs interactions. Notably, the upregulated this one has 24 target sites, while the downregulated this one have 31 target sites. Gene enrichment analysis indicate that the upregulated miRNAs might target the downregulation of genes related to the cell migration and stress responses, while the downregulated miRNAs promote the expression of metabolic genes. Okay, finally, we investigated the regulation by vitamin D via cRNA mechanisms. This network highlighted the interplay between lncRNAs and miRNAs, revealing the downregulated mRNAs are influenced by this LNC and also by this miRNA, which interacts with 21 mRNAs. This interaction downregulates key genes involved in cell growth and metabolism. For upregulated mRNAs, this miRNA emerged as the core regulator, interacting with 27 mRNAs and 2 lncRNAs that are here. And this network enhanced the expression of genes associated with cell proliferation, differentiation, and apoptosis regulation, such as BCL2. Overall, this cRNA illustrates a complex regulatory landscape during Zika infection in vitamin D-treated macrophages. In conclusion, our findings underscore the role of cRNA regulation in Zika pathology, the interplay between miRNAs and lncRNAs influenced by vitamin D, the need for further experimental validation to confirm these regulatory relationships, and the potential for these insights to inform therapeutic strategies. I would like to express our gratitude to the Blood Bank at the Escuela de Microbiología UDA, to all personnel at the institutions involved in this study. Thank you for your attention. I'm eager to answer any questions you might have about our research.